Alright, class, so chapter 4 is all about discussing organic molecules and kind of giving you a little bit of background as to what they are and kind of the cool features that these molecules have. Alright, so we've been talking about different types of atoms and different types of molecules and different types of chemical bonds and how those chemical bonds affect the properties of those molecules. Alright, now we're going to begin to build on that. Alright, so we're transitioning from talking about basic molecules to biological molecules. Alright, so here you're basically being shown kind of an array of biological molecules, which we'll talk about in the next chapter. Alright, but what you'll notice is that every single one of these molecules that you see has something in common. Alright, they're all carbon based. Alright, so when you talk about carbon based compounds, what you're talking about is organic chemistry. Alright, so you can talk about something very simple. Alright, so your simplest organic molecule is something like methane. Right, which I'll show you a picture of in a little bit. Alright, something like a polymer like starch, or cellulose, or glycogen. Right, larger biological polymers. Alright, so organic compounds, right, they contain carbon. Right, organic compounds always contain carbon, but you'll also see some other elements as well. Right, hydrogen is usually one of those, as well as things like nitrogen and oxygen. You'll see sulfur. Okay, but mainly your organic molecules have carbon. Alright, that is the main element in all of organic chemistry. Alright, so kind of give you a brief history on organic molecules. Alright, so it was thought by scientific authorities that there's some other force, right, that's allowing for the formation of these organic molecules. Right? And that this vital force is necessary to create organic compounds in living organisms. In other words, organic molecules don't just spontaneously form out of thin air. That's what was thought. Alright? That they're going to be produced by living organisms. Alright, now this prevailing thought at the time has actually been disproved. Alright, so vitalism is what it's termed. Alright, so it was disproved through a series of experiments. Alright, so here, alright, you are looking at a setup of an experiment that was conducted. Alright, so this is the Stanley-Miller experiment, so around the 1950s, 1953. Alright, so what we have here is Stanley Miller is trying to synthesize organic molecules from inorganic molecules. And what he's trying to do is he's trying to simulate Earth's early atmosphere, right, when the Earth was very, very young. Right, so as opposed to today, oxygen wasn't very readily available in the atmosphere. All right, but there were other substances, molecules, compounds in the air, things like methane, all right, and ammonia and hydrogen, carbon dioxide, things like that, carbon monoxide. All right, very little oxygen. And so using these as a kind of a pool, all right, he created his own little microenvironment to kind of simulate the Earth's early atmosphere. All right, so what we have here is a small flask that contains boiling water. All right, and as the heat's being transferred from your heating source, a candle, Bunsen burner, all right, you have the boiling of the water. All right, that vapor is going to move up this tube, and it's going to enter this large flask. All right, so in this flask, he has these electrodes. All right, and these electrodes provide a nice little spark. All right, this spark, think of it as kind of a stimulant. It's kind of there to get the whole process going. All right, chemical reactions need some kind of jump to move forward. All right, so here, when these gases, all right, the methane and ammonia and hydrogen gas are sparked in the presence of water vapor, all right, you have the products of the reaction, of the interaction of these elements, and they're basically siphoned off. And as they're siphoned off, they get trapped and samples are removed. All right, because here you have this condenser, so you go from this gaseous state back to the liquid. So you have this liquid in this tube here. All right, it re-enters the flask you have. So Stanley Miller analyzed samples, all right, following this process of simulating Earth's early atmosphere, and he found the presence of organic molecules in the liquid water. All right, so things like formaldehyde, hydrogen cyanide, very simple amino acids. All right, so basically you have inorganic molecules. All right, something that's inorganic giving rise to organic molecules. All right, when you think about organic molecules, you think about biological systems and you think about life. All right, so this is kind of the big thing. All right, so you have something that's inorganic that you consider, non-living, and you're creating organic molecules from this. All right, totally goes against the whole vitalism idea. All right, there is no opposing some magical force in nature forming organic molecules. All right, where you need some living organism to produce organic molecules, you don't. They can form on their own. They just have to have the right circumstances, all right, to form. All right, so here, all right, so Stanley Miller's experiment, all right, you have the abiotic synthesis of organic compounds. All right, so here, abiotic. All right, so this is without life. All right, so this is big. All right, this is monumental, life-changing. All right, so scientists can synthesize organic compounds in the lab from inorganic compounds and molecules. Alright, so there is some kind of mechanistic approach. Alright, so there's this idea that you have the physical laws of nature and chemical laws that govern the natural phenomena that occurs in these processes. Right? There is no outside magical opposing force that's causing all this to happen. Alright, you have the laws of nature that are constricting, defining, and allowing for these events to take place. Alright, so we've been talking about all these different types of atoms, right? And you look at the periodic table, and we've talked about how based on each atom's respective total number of electrons found in its outermost shell, alright, so its valence shell, is going to determine how it behaves. Alright, so the electron configuration of an atom is going to affect how that atom behaves, how two atoms interact with one another. Alright, so basically what we're talking about is chemical bonding. Alright, the types of chemical bonds that form and the number of chemical bonds that form. Alright, so it's all about the number of valence electrons. Alright, so you think about carbon. Alright, and if you look at carbon on the periodic table of elements, alright, you'll notice that it has an atomic number of 6. So this is its atomic number. Alright, so based off this information, you can take your little carbon atom, draw your little first orbital. Alright, you can get your first 2 electrons. So 2 out of 6, you're left with 4. Draw your second little orbital. Alright, so basically, if you draw a nice little Lewis dot structure of carbon, this is what it looks like. Alright, so remember when you're drawing a Lewis dot, that you're only looking at the valence electrons. Alright, so carbon has 4 valence electrons. Alright, so it's very possible for carbon to form 4 covalent bonds. Alright, up to 4 covalent bonds. Alright, to reiterate that point, carbon can form up to 4 covalent bonds for single covalent bonds, not any covalent bond. Alright, single covalent bonds, up to 4 single. Alright, these carbon atoms, alright, we'll talk about how these carbon atoms can be arranged, how they can be strung together in a chain, and how they can interact with other atoms of different types. And this is going to affect the overall shape of the molecule, and is going to affect, ultimately, how that molecule or that compound behaves. Alright, just solely based on carbon binding to these other atoms and what those are. It'll affect the chemical properties of the molecule, it'll affect how that molecule folds in on itself and what it looks like, and then how it behaves. and the type of chemical properties it has. Alright, so here what you're looking at is methane. Alright, so methane is the simplest organic molecule you're going to find. Alright, so here what you have, alright, so bringing back our nice little Lewis dot structure, alright, of carbon, and then you have hydrogen. Alright, well, carbon and hydrogen, well, hydrogen has 1 valence electron, carbon has 4. So that means that you can have up to 4 hydrogens that bind to carbon. Alright, and what you get is methane. Alright, and methane has this nice little tetrahedral shape to it. Alright, the atoms in a molecule are going to sort themselves into certain distances away from one another, and they're going to have this nice little configuration of a tetrahedral. Alright, so here, looking at methane, you have the nice little tetrahedral shape. Alright, so this is what a tetrahedral looks like. Alright, so this is what the molecule looks like from a ball and stick model. Now, if you look at something like ethane, alright, so instead of having 1 carbon, you now have 2 carbons. And each carbon atom now is bound to another carbon and 3 hydrogens. Alright, now this covalent bond, right, that joins these 2 carbon atoms is still a single bond. Alright. And here you have basically 2 tetrahedrals that are basically inverted in on one another. Alright, so you have basically the tip top of a tetrahedral, pointing in on one another. Alright, and you have the hydrogen atoms sticking out on both ends. And then you have ethene. Alright, so ethene, alright, it has 2 carbons, but notice here, you have 4 hydrogens. Alright, so 4 hydrogens. Alright, so what you're left with, that means that the carbon atoms share not 1, but 2 pairs of electrons through covalent bonds. Now the cool thing about this is that as you go from a single to a double to a triple covalent bond, alright, this is going to restrict the movement of the atoms. Alright, so if you kind of look at something like methane, alright, it has this hydrogen atom attached to it. Alright, since this is a single bond, alright, that single bond has the ability to rotate on an axis. It can basically spin. Alright, but here, when you're looking at something like ethane, alright, this carbon atom is attached to the other carbon atom through a double bond, alright, that will only allow it to kind of wiggle and waggle a bit. Alright, notice how the molecule is also in the same plane. Alright, both carbon atoms and the hydrogens are all in the same plane. Alright, so basically if you draw a line right through it, alright, they're all found on the same plane. As opposed to something like ethane. Alright, all the atoms aren't found on the same plane. Alright, some of your atoms are found above the plane, below the plane or going out in other directions. Alright, so double bonds and then triple bonds restrict the movement of the atoms in the molecule even more. Alright, the structure becomes more rigid. Alright, so here, so looking at carbon and electron configurations, alright, so when you're talking about organic molecules, alright, so carbon has the ability to interact with other atoms. Alright, now carbon likes to interact with things like nitrogen and oxygen and hydrogen and sulfur sometimes, and you also see phosphorus in organic molecules. Alright, but these are going to be usually a lot of your normal main players, alright, in organic molecules. Now, right, when you're talking about organic molecules like methane, and these particular compounds, alright, if you look at methane, you'll notice that this carbon atom, that it's bound to four hydrogen atoms. Alright, so basically it's totally and completely saturated with hydrogens. Alright, this is a hydrocarbon. It's carbon and hydrogen. Alright, so compounds composed of only carbon and hydrogen are hydrocarbons. Now, this is cool. Alright, because think about it. Think about things like propane. It's a gas. But it's also a hydrocarbon. Okay, it's a fuel source. Now, cool thing about fats, lipids. Fats are a type of lipid. All right, kind of give you a little bit of a sneak peek of chapter 5. All right, fats are a type of lipid. All right, lipids are a class of biological molecules. All right, but fats are also composed of hydrocarbons. Alright, so here what you're looking at is basically a cross section of human adipose tissue. Alright, so basically you see all these little droplets here and here and here and here. Alright, these droplets contain fat. They contain fat molecules. Alright, and this is inside of a cell. Alright, you'll notice that this cell has a nice little nucleus, but it is jam-packed with fat droplets. And a molecule of fat in this fat droplet. Alright, so there's many fat molecules in this droplet. Alright, so just looking at a single one molecule of fat. Okay. This right here, right, that's only one, what we call a fatty acid chain. Alright, notice that you have a total of 3 fatty acid chains. Right now, you have these 3 fatty acid chains. Now, these 3 fatty acid chains, they are also composed of these black spheres. The black spheres are carbon, and kind of the silver gray spheres are hydrogen. Alright, so this is basically a hydrocarbon. This is basically a hydrocarbon. This is basically a hydrocarbon. Well, something like propane, alright, is a hydrocarbon. Something like these fatty acids are hydrocarbons. All right. Propane we use as a fuel source. All right. As used for like heat or something. All right. But also, you look at lipids, look at fats, they're hydrocarbons. They're made up of hydrocarbons. These are also a fuel source. All right, so it's pretty cool. All right, so here's a little animation on the carbon skeleton, the carbon backbone.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
All right, so your carbon skeleton. Your carbon skeleton is backbone. All right, think about your skeleton. All right, you have your backbone, your vertebrae. You have each of your vertebrae you can think of as carbon atoms. All right, and they're linked end to end, basically. All right, it's kind of the same idea with an organic molecule. All right, now the carbon backbone, it can either be straight, all right, so it can be linear, all right, so it can vary in length, all right, so it can increase in length one carbon at a time. All right, so going from something like ethane with 2 carbons and propane with 3 carbons, you can increase in length. You can also have branching. All right, so basically carbon atoms can rearrange in that molecule to form different arrangements. All right, so here something like butane is a straight chain. All right, but then notice that one of the carbon atoms changes its position, all right, so that it forms like this T shape. All right, so this is what we call isobutane. All right, so it goes from being linear to branching. All right, so it's kind of like a straight limb on a tree, and you have a stem branching off of it. It's kind of the same idea. You can also have double bonds. All right, now your double bonds can actually be found in different locations. All right, so basically you can number your carbon atoms. All right, 1, 2, 3, 4. All right, so this tells you 1 butene. All that's telling you is where the double bond's located, so carbon 1. And then you have 2 butene. All right, so carbon 1, 2, 3, and 4. All right, so you basically have a little carbon 4 here. So that 2 butene is telling you the double bond is found here, in second carbon atom. All right, so double bond position can change. All right, which is going to affect how that molecule behaves. All right, and then you have organic molecules that can go from being linear to being cyclic. Basically, they form ring structures. All right, so this is important because we'll start talking about later on carbohydrates, and carbohydrates will differ in their overall shape and arrangement based on whether they're sitting on a shelf as a solid form of that substance or they're placed in an aqueous solution. All right, you'll have either a linear form or a ring form that predominates. All right, I'm not going to give away the secret yet. All right, you have to kind of stay tuned, and we'll get to that topic a little bit later on. All right, but it's important because it's going to have to do with how these molecules behave in an aqueous solution. And you think about our bodies, it's mainly water, right? So these molecules, how they function, how they behave, it's important to understand what they look like in an aqueous environment. Alright, so we're going to look at next isomers. Alright, so this is kind of a short, brief clip on the different types of isomers.
3 types of isomers are structural isomers, which differ in the covalent partnerships between their atoms; geometric isomers, which vary in arrangement of atoms around a double bond; enantiomers, or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
Okay, so when you're looking at isomers, alright, you have 3 main types. You have structural, alright, you have something called cis-trans. These are what we call your geometric isomers. And then you have something called enantiomers. Alright, so first starting off with geometric isomers, or structural isomers. Basically, your structural isomers. The only difference between these 2 molecules is the arrangement of how these carbon atoms are oriented. Alright, so an isomer or isomers is going to be 2 molecules that have the same chemical formula, but they look different. And because they look different, they behave different. Okay, but they have the same chemical formula. Alright, so here, alright, you're looking at these 2 structural isomers here. Alright, you have a linear form, and then you have a branched form. Alright, so they differ structurally, which is basically how the carbon atoms are rearranged in the backbone. That's the only difference. Alright, your geometric or your cis-trans isomers, it has to do with how your side groups are oriented around a double bond. Alright, so think about cis and you think about trans. Alright, cis, basically it's the same. Trans, think about across, transmigration, movement across something. Alright, so what you do to figure out if it's cis or trans, alright, just basically put a plane down the middle of the molecule and look at these functional groups. Alright, both these X's are found on the same side. Alright, so they're cis. Alright, but trans, they're found on opposite sides. Alright, they are across from one another. Alright, so they're trans. This is a trans isomer. Alright, so you're dealing with the same molecule, the same molecular formula, but due to the orientation of these side groups, alright, they're 2 different molecules in terms of how they behave and how they function. And then you have your enantiomers. Alright, so your enantiomers are going to basically have or be they're mirror images of one another. Alright, so you can basically draw kind of a mirror down the middle of this. Okay. And they're mirror images. So they're kind of like your left and your right hand. Alright, so your left and your right hand are mirror images of one another. Okay, but here's the thing. If you try to overlay your left over your right hand, it doesn't fit perfectly. Alright, your thumb is where your pinky is, and your pinky is where your thumb is. So it's not a perfect fit. Alright, and when you're dealing with an enantiomer, it doesn't matter how you spin or rotate the molecule. These side groups, right, these functional groups, are never going to be in alignment with one another. Right, so looking at these two isomers here, all right, notice that they're isomers because they have the same chemical formula. All right, but what type of isomer are they? Are they structural? Are they cis-trans isomers or geometric isomers? Or are they enantiomers? All right, well, these guys, what difference between the two of them? All right, well, look here. This carbon atom here is found at the end in glucose. But then this carbon atom here is found in the chain, the backbone of the molecule. Alright, so basically they are structural isomers of one another. Alright, so these guys are structural isomers. Alright, so looking at these two. Alright, so again, you have the same number of carbon atoms, you have the same number of hydrogens, you have the same number of oxygen. Alright, so they're isomers of one another, but what type? Alright, well, they're structural isomers, because here, you're looking at this carbon double bond at the end, you're looking at the carbon double bond here in the middle. Alright, they're structural isomers. The backbone of the molecule has been rearranged. Now, let's talk about the important implications of these enantiomers. Alright, so looking at a drug, for instance, something like L-DOPA.
Isomers are compounds that have the same molecular formulas but different three-dimensional structures. The difference between isomers can literally mean the difference between life and death. L-DOPA, shown on the left, reduces the symptoms of Parkinson's disease. Its isomer R-DOPA on the right has no effect on the disease.
Alright, so thinking about these enantiomers, and you basically have what they have here as like L and D isomers. Alright. You could basically think of S and L are the same thing, and R and D is the same thing. Alright, so it comes from the old idea that if you're left-handed, alright, there was something wrong with you. Alright. Basically because people, they would shake hands and they would shake hands with the right hand, and then you have people that shake hands with the left hand and it just wasn't proper. They were left-handed, but it wasn't proper etiquette. Alright, so they thought that these people were sinister. So sinistral, sinister, that's what the S stands for. So they kind of stuck. So S, sinistral, left-handed. So that's where that comes from. Alright, so R and D might be for, dominance, that was the dominant hand, and she used to shake hands, so basically R. Alright, so here you have a drug, something like ibuprofen. Alright, we take it all the time. Alright, but there's only one version of that, one isomer of that drug that's actually going to work. Alright, and that's going to be the S isomer. Alright, now, why is that? Why is the R isomer or enantiomer ineffective? Well, it all has to do with the fact that this molecule, that these molecules and these atoms in this molecule have to be oriented in a certain arrangement because they're going to bind to a receptor in the body. And that receptor recognizes basically the pattern of that molecule. And it only recognizes it in one way. Alright? It's kind of like a key and a lock, basically. Alright, now sometimes it's the L enantiomer that's functional, sometimes it's the R enantiomer that's functional. It's going to vary back and forth depending on the particular molecule you're dealing with. Alright, so like albuterol is used to basically open up the airways if a person has an asthma attack. Alright, so the R enantiomer is what's effective. Alright, so we're going to talk about functional groups. Alright, so far we've talked about how organic molecules, compounds, can vary in their size in terms of the length of the carbon atoms, the number of carbon atoms you have. They vary in their shape. Alright, so you have different arrangement of the carbon atoms. So you can have linear and branched, and you can have the presence of double bonds. You can have these organic molecules that form ring structures. And then they will become even more complex depending on how all the other atoms in that molecule orient themselves, alright, in space. Alright, and get a defined shape. Now, there's another component, alright, to how molecules behave, and that's their functional groups. Now, their functional groups are going to give molecules very key properties. And the functional groups are also going to be important in chemical reactions. Alright, because they can participate in chemical reactions. Alright, so the forming and breaking of bonds. Alright, so we're going to walk through each of the types of functional groups. Alright, so you're going to want to familiarize yourself with the name of the functional group, what that chemical group looks like, and its key properties and characteristics. Alright, so the first one, your hydroxyl groups. Alright, so your hydroxyl groups have an OH. Alright, so hydroxyl groups are basically what make up our alcohols. Alright, so things like ethanol, or something like methanol, or something like propanol, or something like isopropanol. Alright, so when you're looking at alcohols are polar. Alright, so again, think about what this word means in terms of polar. You're dealing with an atom that is highly electronegative. Well, things like nitrogen and oxygen and phosphorus and fluorine, these are all highly electronegative atoms. Alright, so here, your oxygen atom that is attached to the hydrogen, alright, is also attached to the carbon atom. All right, it's basically pulling electrons from the hydrogen. All right, and it's also pulling electrons from that carbon atom. All right, so it's sharing electrons with these atoms, but the oxygen atom is, in this case, more electronegative than hydrogen and more electronegative than carbon, and more so in the sense that basically the covalent bond, all right, that joins together the carbon and the oxygen atom, here is a polar covalent bond, and then the covalent bond that joins the oxygen and hydrogen is also polar. Alright, so what that means is that this OH group is a polar functional group. Alright, so that means that your alcohols are soluble in water. Alright, so they can form hydrogen bonds with water molecules. Alright, so the next group are your carbonyl groups. Alright, so your carbonyl groups, you basically have this carbon atom that's attached to an oxygen through a double bond. Now, there are two types of carbonyl groups. Alright, there are ketones and there are aldehydes. Alright, so a carbonyl group is referred to as a ketone if the carbonyl group is found within the backbone of the carbon skeleton. Alright, so let's look at acetone. Alright, so acetone here, notice that you have this carbon atom attached to the double bonded oxygen. Alright, this guy here. Now, since this is found within the backbone structure of the organic molecule of acetone, this is a carbonyl group that's referred to as a ketone. Alright. Now, looking at an aldehyde, which is another type of carbonyl group, this is where the carbon double bond of the oxygen is found at the end of the molecule. Alright, so looking at propanol. Alright, so looking at propanol, here you have your carbon double bonded oxygen at the end. This is an example of a aldehyde. Alright. So, particularly, we'll talk about sugars later on. Alright, but your sugars, alright, when we talk about carbohydrates, your ketone sugars are referred to as ketoses, and your aldehyde sugars, aldoses. Okay, so let's take a step back here. All right, so look at, for instance, glucose and fructose. All right, well, these two molecules are isomers of one another. All right, have the same chemical formula, but they differ based on their functional group, where the location of that functional group is. All right, so glucose, since this carbon double bond oxygen is at the end, all right, this is a aldehyde, which makes this an aldose sugar. All right, and then looking at fructose, since this is found within the backbone of the skeleton, this is a ketone, which makes it a ketose sugar. All right, so our next functional group is a carboxyl group. All right, so a carboxyl group is also referred to as a carboxylic acid or organic acid. All right, so we've come across this particular functional group in class. Alright, so we've talked about acetic acid in class. Alright, so when you're looking at carboxyl groups, alright, notice that you have carbon atom joined to the oxygen. Well, here your oxygen atom is pulling electrons to itself. Alright, so here you have your carbon atom joined to the double bonded oxygen, oxygen, hydrogen. Alright, basically you have all these electrons, alright, that are being pulled to this oxygen atom. Alright, because oxygen is more highly electronegative. You have all these electrons that are being pulled towards this oxygen because it's more highly electronegative than carbon. And then you have more electrons being pulled to this oxygen because hydrogen is has a very low level of electronegativity in comparison to oxygen. Alright, so the oxygen atoms are highly electronegative, so they're pulling all these electrons to them. Alright, so this makes carboxyl groups very polar. Now, your carboxyl groups also are acids. Alright, so again, what is an acid? Well, an acid has the ability to donate, release protons. Alright, and recall that only free protons can affect the pH of a solution. Alright, so here you have your non-ionized carboxyl group. Alright, and in here, once your hydrogen ions are released, you end up with a ionized carboxyl group. Alright, so that means that this oxygen down here gets a nice little negative charge, and you start plopping off free protons, which can make the pH go down. Alright, become more acidic. Now, the next group, your amino groups, or your amines. Your amino groups, alright, again, are going to be polar. Alright, so here you have a nitrogen attached to a hydrogen attached to another hydrogen. Alright, now again, nitrogen is more electronegative than hydrogen. Alright, so let's pull electrons to itself. Alright, so it gets a nice little slight negative charge. Alright, so this makes this little group a nice little polar group. Alright, now looking at this molecule of glycine, notice that it has an amino group. Now, your amino groups can act as a base. Alright, now bases, they pick up protons from the surrounding solution. Alright, so here you have your little amino group that picks up an extra proton and now is ionized. Alright, so it ends up with a full positive charge on the nitrogen. Alright, so that means that, for instance, you could end up within a cell. Alright, here you have a molecule of glycine. Alright, now later on we'll talk about proteins and we'll talk about how proteins are made. Alright, but kind of give you a little bit of a preview. Alright, glycine is an amino acid. Alright, so amino acids are building blocks of proteins. So think about this word, amino acid. All right, so it's an acid because it has a carboxyl group, carboxylic acids. The word amino comes from the fact that it has an amino group. All right, every single amino acid is always going to have a carboxyl group and an amino group. Okay, now in a cell, your little bitty amino acids can actually ionize. All right, at a given pH. All right, so what that means is you can end up with an amino acid like glycine. All right, which is one of your simplest amino acids. It gets a nice little negative charge. Okay. And then your little nitrogen here gets a nice little positive charge. When it picks up another hydrogen atom. Alright, so this would be the ionized form, alright, of this particular amino acid, glycine. Alright, so your sulfhydryl groups, or your thiols. Alright, so this is a sulfur bound to a hydrogen. Alright, now, sulfhydryl groups. These guys are important because, again, you have particular chemical groups, right? Particular things like amino acids, for instance, things like cysteine. All right, cysteine is another type of amino acid. Again, look here. You have your amino group, you have your carboxylic acid. All right, now your amino acids, all right, they're always attached to a central carbon. All right, they're going to have a hydrogen atom. All right, so this basically makes up the overall basic structure of an amino acid. All right, boom, this here. Every single amino acid looks like this, except for what we call its own functional group. All right, this little guy right here. All right, this guy right here is what we call the R group. All right, so this R group basically determines how that amino acid behaves. It basically determines what the amino acid is. All right, because every single amino acid looks the same because it has this same basic structure, and the only thing that's going to vary between them is their R group. All right, you could switch out R groups and you get different amino acids. Now, what's cool about sulfhydryl groups? Well, sulfhydryl groups can interact together, and they can basically cross-link through their sulfur atoms and form stable covalent bonds. And so when you're talking about proteins, you can have the formation of covalent bonds between two different cysteine amino acids in different parts of the protein. All right, later on we'll talk about protein structure. All right, and this is going to be important later. All right, your phosphate group. Your phosphates are also polar. All right, so notice here, look at this. You have oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus, oxygen. All right, so basically end up with this big polar functional group. All right, that ends up with this negative charge at the end of the molecule. So something like glycerol phosphate. All right, that makes this end of the molecule very polar. Now, your phosphate groups. Your phosphate groups are an important component of basically DNA, things like RNA, things like ATP. Alright, so you're basically talking about your nucleic acids. Now, we'll come back to phosphate groups. Alright, so here it talks about phosphate groups having the potential to react with water to release energy. Alright, we're going to come back to and talk about ATP in just a little bit. Alright. Your methyl groups. Alright, so carbon atom attached to 3 hydrogens. Alright, so CH3. Alright, your methyl groups are going to be involved in basically gene regulation. Basically, they're involved in determining what genes are turned on to be transcribed and translated, and what genes are going to be turned off so they're not transcribed and translated. Alright, this is also important when you're talking about DNA replication. Right, you get something that's called hemimethylation. Alright, where basically your old DNA strand is going to be methylated. Alright, but the new strand, alright, for a period of time is not. Now the reason being is because that old strand serves as a template, right, for the synthesis of a new strand. And so you have basically a repair mechanism, a kind of a screening process, whereby the cell will go back in and check that newly synthesized strand of DNA and make sure there are no mistakes. And so the way the cell distinguishes between what strand of DNA is new and old is whether or not it's been methylated. Alright, so for a short period of time, the new strand of DNA is not methylated. And after it goes through this checkpoint, it's methylated. Alright, so try to identify what functional groups you see in the following molecules. Right, so what's present? Alright, well here you have a carboxyl group. Alright, what about in this other molecule? Alright, you have another carboxyl group, but then you also have a carbonyl group. Specifically, you have a ketone. Now, if you try to compare these two molecules, alright, which of the two is the stronger acid? Alright, A or B? Well, the answer is B. The reason being, alright, you think about what an acid is. An acid has the ability to give up protons, right? Well, here, look, you have a carbon atom attached to double bonded oxygen, carbon bound to double bonded oxygen, carbon attached to a oxygen attached to hydrogen. Alright, so you have all these electrons that are being diverted to the oxygen atom, and so that makes this oxygen atom's hold on this hydrogen very weak. Right, so it can very easily be plopped off and you release protons. Alright, so what functional groups do you see in this molecule? Alright, well here we have a carboxyl group. You have a hydroxyl group. Alright, so we have carboxyl. We have a hydroxyl. Then we have an amino group. Alright, so this is something you should be able to do. Alright, you should know what functional groups look like, you should be able to name them, and you should be able to find them, and you should be able to describe the properties of each of these functional groups and what those properties will give a molecule. Now, why am I spending so much time going over functional groups? Right. Well, when you look at a functional group, all right, they are going to have a profound effect on the overall properties of that biological molecule. All right, so here you have two hormones. You have testosterone and estradiol. Now, both of these molecules are what we call steroids. All right, steroids have this very characteristic four-ring structure. All right, you notice that they have the same basic structure. All right, compare testosterone and estradiol; they look very similar. But why exactly are they different? All right, what makes the two chemicals different from one another? Well, it's the functional groups they have. All right, testosterone, you have a methyl group, and you also have here, you have a carbonyl group. All right, specifically a ketone right here. And then when you're looking at, for instance, estradiol, you have your hydroxyl group. All right, so these very slight differences in functional groups aren't going to have an effect on whether you have testosterone or estradiol. So whether you end up with a male or female. Now, coming back to this topic of ATP. All right, so ATP is adenosine triphosphate. All right, so adenosine triphosphate, or ATP, all right, it is a type of nucleic acid, and ATP is basically the currency of the cell. All right, so think of it as money. All right, so when you look at ATP, notice you have this adenosine, and you have this triphosphate. All right, so you have three phosphate groups. Now, notice, these three phosphate groups really don't want to be attached to one another. All right, so this is very, very, very unstable. All right, so ATP doesn't usually stay in this form for very long. All right, it usually is going to be broken apart, all right, to release energy. All right, but notice you have phosphate group with negative charges attached to another phosphate group with a negative charge, attached to another phosphate group with a negative charge. What happens when you get a negative charge and a negative charge next to one another? They repel one another. All right, so there is inherently a repulsive force in these chemical groups. All right, they don't want to be attached to one another. All right, but this is good because this increases the amount of potential energy stored, all right, in this biological molecule. Now, what you can do is you can add water. You take water here, and you can break this phosphate group. All right, so phosphate at the very end of ATP is the one that's broken first. All right, this is the one that contains the most energy. Alright, so one at the very end. Alright, that covalent bond that joins the phosphate to the phosphate group. Alright, when that gets broken, when it gets what we call hydrolyzed, you end up with this inorganic phosphate all by itself. And you end up with ADP, 2 phosphates. Adenosine diphosphates. And then since you're breaking chemical bonds, guess what? You're releasing stored energy. Alright, and this stored energy can be used to do work.